Dear students, my name is Avinash, lecturer in the Department of Electronics and Communication Engineering, Jain Polytechnic, Belagavi. Okay, in this session, we are going to study fourth unit of medical electronics subject, that is measurement and analysis technique. In this particularly, that is we are studying different cell counting method. Okay, so let's see what are the contents we have covered in this session. The contents covered here are introduction to cell counting, methods of cell counting, means what are the different methods are there for the cell counting, optical method cell counting, and last one, the Coulter's method of blood cell counting. Okay, these four concepts we are going to cover in this session. Okay, so let's start with the introduction to the cell counting. So dear students, all we know, that is changes in the normal functioning of an organism are oftenly accompanied by the changes in the blood cell count, right? Okay, so usually the determination of the size of the blood and the number of cells is very important to diagnose a disease, because all we know, the blood constitutes around 5 to 10% of the total body weight, and an average adult having 5 to 6% of 6 liters of blood. Okay, and this blood is made up of composed of white blood cells, RBCs, and platelets. Okay, so we have to follow a different counting methods for these blood cells. That is the most common and routinely applied method of counting blood cell even today, particularly in small laboratories, is the microscopic method. Okay, in which diluted sample is visually examined and cells are counted. Okay, now commonly this is known as counting chamber technique. Okay, it suffers from several common drawbacks. Okay, for example, apart from inherent error of the system, which may be about 10%, there is an additional subjective error of 10%, which may be a plus 10% or minus 10%. You can say also it is a tolerance of entailing the poor reproducibility of results. Okay. Furthermore, the lengthy procedure involves the result in the rapid tiring of the person making the examination. Okay, it's not a fast process. Okay, there are also poor time and labor utilization. Okay, so hence this microscopic method having a different drawbacks, we are going to study the different methods of cell counting. So let's see what are those different cell counting methods in the next slide. So in this chapter we are studying the two methods of cell counting, and that first one is optical method cell counting, and second one Coulter's method of blood cell counting. Okay, in optical method, obviously we are going to use the help of light to count the blood cells. Simply a light is made to fall on a blood sample, and how much the light is scattered, we are going to calculate the blood cell based on that. And second one is a Coulter's method. Here we are using Coulter's counter to count the number of blood cells present in the sample. Okay, so let's start with the optical method of cell counting. Moving to the next slide, that is optical method of cell counting. So we start with the optical method of blood counting. So this method is based on collecting the scattered light from the blood cell and converting it into the electrical pulses for the counting. As shown in the figure, the blood is taken and it is put in the sample for the optical detection system. Okay, that sample of blood is dilute blood. Okay, that is taken in a glass container. Okay, it is drawn through a counting chamber in which blood stream is reduced in a cross section by concentrating high velocity liquid set. Okay, a sample optical system provides a dark field illumination zone on the stream, and the light scattered in the forward direction is collected on the cathode for photomultiplier tube. Okay, the pulses produced in the photomultiplier tube corresponding to each cell. Okay, these signals are amplified in a high input impedance amplifier and fed to an adjustable amplitude discriminator. Okay, the discriminator provides pulses of equal amplitude which are used to drive the digital display. Okay, the instrument based on this technique takes around 30 seconds for the completing the count. Okay, and accuracy of 2% is attainable, means 98% it is gives accurate readings. Okay, the instrument requires around 1 milliliter of sample of blood. Okay, this is about optical method of blood cell counting. Okay, now moving to the next method, that is Coulter's counter, or you can say that is next one is that is Coulter's method of blood cell counting. Okay, before going to that Coulter's method, so I have listed here the procedure that what each of that part will do. So the method is, as I told, the method is based on the collecting the scattered light from the blood cells and converting into the electrical pulses. That around 1 ml of blood sample is taken, and this blood sample is, when it is kept in a glass container, that light is made to fall on that, and how much the scattered light will scatter, that is collected by the photo detector and then multiplied with the help of that photo multiplier. Okay, and that pulses are amplified with the help of amplifier, and with the help of that discriminator, amplitudes are adjusted. Okay, this is the optical method of cell counting. So now move to the last concept, that is Coulter's method of blood cell counting. Let's start with the next method, that is Coulter's method of blood cell counting. Okay, a wide range of particle counting instrument designed to meet a wide variety of needs in the hematology laboratory are being commercially produced. Okay, these instruments range from small counters used primarily for red and white cell counts in very small hospitals and clinics, okay, to the multi-parameter microprocessor controlled instrument featuring fully automatic diluting of samples and printing of results. Okay, so this figure on the slide shows a block diagram, shows a principle of Coulter counter. Okay, a platinum electrode is placed inside the orifice tube, and a second electrode is submerged into a beaker containing a cell dilution. Okay, this will create an electrical circuit between the two electrodes. Current will flow from one electrode to the another electrode through this orifice. Okay, when the cell suspension is drawn through the orifice, cells will displace their own volume of electrolyte and cause the resistance change, which is converted to an voltage change, and it is amplified, and then this is displayed. This is the basic principle. Okay, in practice, the cell suspension is drawn through a orifice by means of a mercury manometer. That manometer includes two platinum wire contacts. You can see A and B as shown in that diagram. You can see that set through a glass walls. Okay, contact A will start the count and contact B will stop the count when it is precisely 0.5 ml of dilution has passed through this orifice tube. Okay, thus it provides a count.